Today, we're going to learn about extracellular ATP. So, what is ATP? Let's start with this guy. His name is Jim. Let's take a closer look at Jim. Under his shirt is his arm, and in his arm there are cells. Jim is made up of countless cells that need energy in order to keep Jim alive. In eukaryotic organisms like Jim, energy is synonymous with the nucleotide adenosine triphosphate, also known as ATP, which is produced in the mitochondria via glycolysis and oxidative phosphorylation with ATPases such as ATP synthase, which are all coded by a variety of genes such as ATP5A1, ATP5B, MTATP6, and so on, and it is produced 24/7, 365. Historically, a lot of what we know about ATP was discovered around the same time in 1929. The man credited with the first discovery of ATP is Carl Lohmann in Germany. However, its function was also discovered independently in Harvard Medical School by Yellowbrigada Sbarro. A team of Drury and St. Georgi were the first credited in finding extracellular nucleotides such as ATP in their work that focused on the physiological activity of adenine compounds in 1929, while its actual structure was discovered in 1948 by Alexander Robert Todd. So in the end, ATP keeps cells alive and healthy, right? Well, sometimes something happens to the cells that can cause the cell to undergo necrosis or unintentional cell death and release all their contents to the environment within the body. This could be due to injury, infection, or great stress to the cell. So let's say, while we were talking, Jim accidentally gave himself a paper cut on his finger. This would mean that he broke some of his cells, causing them to release their contents, such as ATP, into the extracellular space. So now Jim's finger has a bit of extracellular ATP floating around. The main function of extracellular ATP in regards to the immune system is that it mediates the permeabilization of macrophages. Immune cells recognize ATP that is released from damaged tissues and dying cells as a danger signal that elicits a variety of inflammatory responses. Following its release into the extracellular space, ATP is rapidly hydrolyzed in a stepwise manner to ADP, AMP, and adenosine by ectonucleotidases. This process terminates P2 receptor activation, prevents receptor desensitization, and sets the stage for rapid and repeated signaling through purinergic inside-out signaling via purinergic receptors. In addition to these mechanisms, Adenosine Kinase can convert extracellular adenosine into two different adenosine deaminase isoforms called ADA and ADA2 or CECR1, which are found on the surface of many cell types including leukocytes. Both isoforms promote co-stimulatory signaling at the immune synapse, resulting in increased production of T helper 1 cytokines and increased T cell proliferation. The importance of ADA is further underscored by the fact that a genetic disorder resulting in ADA deficiency causes severe combined immunodeficiency, also known as SCID, a condition characterized by impaired T and B cell development and an increased risk of infection. Treatment for this usually involves a bone marrow transplant. The opposite of this is not too great either. Too much extracellular ATP for whatever reason can cause an increase in DNA fragmentation, as can be seen in a deoxynucleotidal transferase assay. This can also cause an increase in protein levels of the tumor suppressor p53, which is currently thought to regulate apoptosis. Both of these can cause a glomerular disease, which means messenger cells undergo cell death by both necrosis and apoptosis. The symptoms of this disease include hematuria, or blood in the urine, reduced glomerular filtration rate, which means inefficient filtering of wastes from the blood, hypoproteinemia, meaning low blood protein, and edema, which means swelling in parts of the body. Treatment for any glomerular disease is aimed at protecting kidney function by reducing proteinuria and controlling blood pressure. Wait, so hold on. This process needs to be stopped somehow, and luckily there's a nice little feedback loop that goes on, as extracellular ATP and adenosine are recognized by the chemotactic and phagocytic reactions in microglia through P2Y receptors. The amount of ATP and adenosine decreased in the extracellular space, slowing down the processes that use it, effectively being its very own off signal. So, this leaves our friend Jim here with little to worry about when he gets a small cut, as the risk of infection is decreased thanks to the signaling of extracellular ATP. Next time, be more careful, Jim.